Good morning, everyone. Today I welcome you all to my defense of my Ph.D. thesis, and I feel quite honored to be the first IC student of the first batch to welcome you all to this defense year. The topic is not-one-dependent suppression of interleukin-6 mediated STAT3 activation, and now in the next few minutes I would try to take you all through a brief journey of the small story about receptors in the JAK-STAT signaling cascade. So, to start with, what are the nodes? What are the NLRs? These NLRs are classically defined intracellular pattern recognition receptors. These have been defined in a way that they recognizes different breakdown peptidoglycan components and elicit an inflammatory immune response. They have been also defined in a way that they interact with the different components of the apoptosis regulatory cascades and lead to the apoptosis signaling in a particular cell. And these NLRs have been classified based on their effector domains as such, via which they bring out their respective. So far there have been a lot of progress in this field of NLR signaling as such, and as more as 23 members of these NLRs have been defined in the human genome as such. So to have a brief look at these NLRs are classified as having a card or the gas-based activity recruitment domain. These NLRs via these card domain interact in card-card interactions with similar downstream kinases having card domains, or having the pyrene domain via which they bring out their gas-based recruitment activities as such. Interestingly, they also have a leucine-rich repeat domain, which is more or less a constant in all the NLRs, along with the central NACT domain. So what these two domains do is, like, this LR, or the leucine-rich repeat domain of these NLRs, is defined as the region responsible to recognize their respective ligands upon which the activation of these NLRs gets triggered, and the central NVD domain, the NAC domain, is having an intrinsic APase activity which actually regulates the oligomerization or the autoactivation process of these NLRs, and then they tend to oligomerize and interact with their respective downstream partners as such. If you see among these NLRs, there is NLRP10, which do not have a leucine-rich repeat domain, and also interestingly there is NLRP1, which has both a pyrene and a card domain. These different architecture of these varied NLRs are constantly being investigated and being held responsible for their varied functions in different immunological pathways as such. And though these NLRs were started as being defined as intercellular receptors for peptidoglycan recognition, their field of investigations evolved into various different aspects. And if we see here, this looks quite complicated, and in fact it is. because these NLRs have been defined being involved in various cascades. For example, NLRP 5, 14 and 7 have been involved in many genetic disorders as such. The ninth NLR, it is classically involved in the central nervous system disorders as such. If you see NLRP3, it is very well defined nowadays as being involved in regulating inflammatory responses mediated via interleukin 1 as such. And if you see the NOD 1 and NOD 2, the mutations in these NLRs have been defined to be responsible for the genesis of many inflammatory diseases like the Blau's syndrome, or also being involved in asthma as such. So as these NLRs are being now investigated into various diverse aspects, what we thought is we would go back to the classical NLRs NOD 1 and NOD 2, who were the initial NLRs being investigated, and what we thought that we would investigate the role of these nodes in slightly different aspect of the immunological system, then as such as like the new NLRs, which especially the NLRPs, which are being investigated nowadays. So in the literature, NOD 1 and NOD 2 are actually being defined to recognize different breakdown peptidoglycan components, and upon recognition their oligomerization takes place, and when they oligomerize, they lead to the activation of the NF-kappa B essential modulator, which actually results in the release of NF-kappa B in the activated form, which translocates to the nucleus and results in the release of pro-inflammatory cytokines. So taking this picture into concern, what we thought that why not take out the NLRs NOD 1s in special and investigate its role in what we will show, the JAK-STAT signaling cascade. So why JAK-STAT signaling cascade? Now JAK-STAT signaling cascade, as we all know, is a very important signaling cascade in the immune regulation, and disorders with the JAK-STAT signaling cascades have been depicted in many physiopathological conditions as such. For example, progression of many malignancies, neuromuscular disorders, cardiomyopathies, and so on. So what we were interested is to investigate the role of NOD 1 in the JAK-STAT signaling cascade. So to start with, what we initiated was overexpression studies in HEC293 cells, and we tested different cytokine and STAT combinations, for example, interleukin 6, STAT3, alpha and gamma, STAT1, and so on. And what we observed in these very different combinations is that the effect of NOD 1 or interleukin 6 mediated STAT3 activation was most prominent, and the prominence is that with overexpression of NOD 1 we see a dose-dependent clear inhibitory effect of both basal and interleukin 6 mediated STAT3 activation. And in contrast, in comparative setup, what we observe is that under similar experimental conditions, the overexpression of NOD 1 results in no significant effect on both the basal or alpha and gamma induced STAT1 activation as such.